So the emphasis in the AP curriculum used to be on the animals, specifically the mammalian body, and how the systems work together. A lot of the focus really now is on the cellular level. However, we do really need to, in order to understand muscles and muscle cells, we do want to look first at kind of the big picture. So when we look at the animal body, we do see a variety of cells. So this is where, of course, cell differentiation is going to come into play, and the idea that not all cells are the same. And when we look at the animal body, we are going to focus a little bit on locomotion, and or transport. So we are actually going to see transport both within the body and to allow the body to move through space. So because there is a huge advantage for any organism that can move through space, we are going to see a variety of structures that allow this to happen. So what we see on a unicellular level, which is not an animal, is we see the ability to move your cell. Move your cell. This is the cilia, this is the flagella, this is the cytoplasmic streaming of the yeast that we looked at, the schmooze. This is the amoeba being able to move. But when we look at the actual multicellular organism, this is where we start to see cell differentiation and the unique ability of some cells to help produce whole organism locomotion. So cell differentiation is really big, but of course we also have to be able to communicate. We don't want the muscles contracting at different times. We want the muscles contracting in response or as a purpose. So what we're really going to look at is how do the cells differentiate and communicate in order for this to happen. One of the unique things that we'll see is that muscles and bone are going to be important for locomotion depending on the environment. So we do see the ability to move on land, on air, and in water. And even though the environments are different, we do actually see a similar structure. So all muscles are actually going to have a similar structure, which is called a sarcomere. So this leads us to believe that the sarcomere evolved before animals diverged into these different habitats. So the idea that the sarcomere is really conserved. However, not all organisms that move actually have bones. Joints are composed of bones at an intersection or a joint, and the muscle moves the bone. And that is what really gives you locomotion, is the muscle contracting and moving the bone. Now there are 3 different types of muscle. We're not going to go too much into them, except to know that smooth muscle is involuntary. This is what's going to be in the intestines. Skeletal muscle is mostly voluntary. And then cardiac muscle is actually going to be pretty unique, and we don't have to get too much into that, but we do want to know that there are varieties of muscle cells. So even though the basic unit is the same, we still even see some differentiation occur. So now we want to get into what is the actual muscle cell and how does a muscle contract. So we do need to look first at the structure. So a muscle cell, if you scroll down and look at the next picture, way down here, a muscle cell is actually very, very long, and it has multiple units called a sarcomere. So a sarcomere is the basic unit. So we usually think of cells as being microscopic, but muscle cells are actually macroscopic. You can actually see the cell. It is the basic unit of muscle in a cell. And what a muscle really just is two types of protein. So both actin and myosin are proteins. Well, we know that proteins are made of amino acids. And so what it turns out is that the muscle cell is going to build proteins in a specific structure and then actually going to contract those proteins. So the way that a muscle contracts is called the sliding filament mechanism of contraction. But here's where we have to be careful. The proteins don't get shorter. What really happens is this idea of sliding. They actually slide closer. So we don't have to memorize it anymore, but you used to have to be able to identify different parts of the sarcomere. You used to have to be able to identify the Z-band and all of these different things. We don't have to do that anymore because what we're focused on now is what is the idea of muscle contraction? And how does a muscle know how to contract, and what does this look like on a cellular level? So the actin is actually going to be the base protein. So the actin is a thin protein, and myosin is arranged into what are called heads that bind to the actin. And so what happens is this idea of almost reaching out and pulling back. The myosin heads are going to conformationally change. So the actin does not really ever change shape. The myosin is the part of the protein that is actually going to do the work. So of course, this is where we'll see ATP in order to do work. So the actin really stays in place, but as the myosin pulls, this will shorten in. And that's what a muscle contraction is actually the decreasing of space. So how does this happen? How do we make these proteins come closer together? What it has to do with is the ability of the myosin head to break ATP apart. So ATPase is the digestion of ATP into ADP plus an inorganic phosphate. Now, of course, we know what the source of ATP is. So you should be able to talk about where does ATP come from, how do I get ATP, both glycolysis, the Krebs cycle, and the oxidative phosphorylation. What we have to pay attention to on the cellular level is what is ATP actually doing? We say that it's energy, but what energy? What does that mean? What is it actually doing? So what ATP is actually doing is we are moving this myosin head. So when we look at the smallest structure of a muscle, we see actin and we see myosin. And the myosin will bind to the actin. The myosin will change shapes, move the actin, and then the myosin will release and will slide back. So every muscle contraction is this cycle. Bind, move, release. Bind, move, release. So let's look at how that actually happens. So the resting state is actually when the muscle is released. So down here, this is the resting state. And what has to happen in order for a muscle to contract is for ATP to be broken down. So when ATP is broken, ATP hydrolysis, this is going to allow a shape change or conformational change in the protein. So ATP is there, we're relaxed muscle. When ATP is hydrolyzed, then there's a shape change and the myosin will bind, or it's called a cross bridge, to actin. As it binds, we are going to cause a conformational shape change. So after binding, you have a second shape change. This second shape change is going to cause you to release products, which are ADP and inorganic phosphate. And now I've released it. And in order for this muscle contraction to stop, so to stop contracting, I need ATP. So if there's no ATP, the muscle is actually permanently contracted, which is why when someone dies, they have what's called rigor mortis, which is where their muscles are permanently contracted. We also see some disorders such as botulism, which is where all the muscles are relaxed, or we see the opposite, which is tetanus, which is where all the muscles are contracted. So that's going to be happening here on a cellular level. So what you need to know from this picture is you need to be able to identify the actin, the myosin, and the role of ATP. So when ATP is bound, the muscle is relaxed. ATP should get broken down, allow the myosin head to bind. So ATP is broken down. A myosin can bind to the actin. Once it binds, it will cause a contraction or a shortening, bringing the parts a little closer together. And then ADP gets released once that shape change, that second shape change is done. So now I'm in a contraction, and in order to release it, I need more ATP. So what is it that is actually able to control this process? How do we regulate muscle contraction? Down here at the bottom is a picture of the sliding filament theory. It does show you the I band, the H zone, the A band, the sarcomere. Basically, you just need to know the components, the actin, the myosin, and the idea that it's not actually physically shortening the proteins, but bringing them closer together, which is why we call it the sliding filament. The way that we regulate or control muscle contraction first is the presence of ATP, and we've actually already covered what that means. But it turns out that there are some other components. The first is tropomyosin. Tropomyosin is a protein that binds to the actin and blocks myosin. So the way that we keep muscles the way that we control whether this is happening is we keep muscles in a relaxed state by blocking the myosin head from binding. And so that's what tropomyosin does. Troponin is another protein that holds tropomyosin in place. So basically you're blocking myosin from binding to actin. If we want the muscle to contract, we need a signal. The signal here is actually calcium. Calcium is going to be a signal that removes troponin and loosens tropomyosin. So when we're ready for the muscle to contract, calcium is released. Calcium binds to troponin, removes troponin. Tropomyosin is loosened, and the myosin can now bind and perform the sliding filament. So how does calcium know how to be released? Well, if we think about how we contract a muscle, the muscle contraction relies on a nerve signal. Nerves are what tell the body, control the body. And so what's going to happen is that acetylcholine is released from a nerve. And we'll look a little bit closer at this. Once acetylcholine is released, acetylcholine will actually signal for the release of calcium from the sarcoplasmic reticulum, which we'll look at